Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guest today is Rachel Haurwitz, co-founder, president, and CEO of Caribou Biosciences. Caribou is a CRISPR genome editing company spun out of Jennifer Doudna's lab at the University of California, Berkeley. In this episode of Lab to Startup, we will talk to Rachel about how she built Caribou right after finishing a Ph.D. in Jennifer's lab and went all the way to taking this company public last year in the midst of a pandemic. We will talk about how she took a very nascent idea called CRISPR, built a team around it, raised funds, built collaborations with big companies, and built this very successful company that has recently reported phase 1 clinical study results. I've been fascinated by Rachel's journey and been waiting a long time to bring her on as a guest on the show. So please enjoy my conversation with Rachel Haurwitz. Rachel, welcome to the show. I was wondering where to start this conversation, and I think the best place is to have you walk us down the memory lane to the beginning of your startup journey. Even before you incorporated the company with Jennifer Doudna, your advisor in grad school, when did this idea of startup come up actually? I read somewhere that you wanted to become a patent attorney. I'm so glad you didn't take that path. And also, all this happened before Jennifer Doudna won a Nobel Prize, and when no one imagined that CRISPR would be such a game changer, especially because gene editing has been happening for a long time with not much success in solving human diseases. So what made you go down this path?
Naresh, thank you so much for the invitation today. So winding the clock back more than 10 years, I was the first graduate student in Jennifer Doudna's lab to work on CRISPR. So scientifically, very much right place, right time. And I had the chance as a PhD student to really dig in on a lot of esoteric biochemistry and structural biology, starting to understand some of the moving parts of these really interesting bacterial immune systems, part of this large family of CRISPR systems. As we got started on our work, clearly this turned into a really interesting area of science. And pretty quickly, there were many, many people in the Doudna lab who were working on many different aspects of these elements of biology. It started to become clear that not only was this incredibly cool biology in terms of how bacteria or archaea could use these systems to fight off viruses, bacteriophage, or other selfish genetic elements, but it also became clear that there were many ways that you could start to repurpose some of these enzymes for applications. Now, at the time, her laboratory was a place that was really a training ground for the next set of professors at your favorite university. I was one of a very few people who came in, you're right, thinking I wanted to be an attorney when I grew up, and specifically not wanting to be a professor when I grew up. So I think I had a different viewpoint on these exciting new technologies. So she and I started having a lot of conversations that were very general in scope initially about how cool the science was, how promising the new technologies could be, and what one hypothetical team might be able to do in the future. And that eventually morphed into, well, maybe we are that team. Maybe we can build the company that can start to tackle some of these opportunities. And that's what we did. So towards the end of my PhD, she and I hopped on BART together, went over to San Francisco, sat in a beautiful conference room in a law office downtown, and incorporated Caribou with a really big vision for how we could bring some of these really exciting, nascent, new CRISPR-based technologies into a company to try to really push them forward.
And I'm guessing this was your first experience in a startup. How hard was it for your transition from your grad school to a startup straight into it? having no experience. Talk about that decision-making process.
Not only is this the first startup that I've been part of, it's actually the only company I've ever worked for. So it was an incredibly steep learning curve for me. I fundamentally went from my job being pipetting clear liquids from one tube to another to my job being building and growing a company, setting strategy, hiring a team, and fundraising for it. I continue to learn every single day on the job, which makes this an incredibly exciting place and space to be. I think that being in the Bay Area helped to normalize what a huge risk I was actually taking. We live in an ecosystem where there are actually a lot of people who are in their 20s who found companies and go on to do really interesting things. And I think it's also a space where people found companies, they fail miserably, you learn something from it, you wear it as a badge of honor, and you move on to the next opportunity. So it helped a lot that at the time, some of my closest friends, not in biotech, but in other industries, were actively also founding companies at the same time. And we could compare notes on a lot of the building and growing pains that were shared across different industries. Certainly for me, getting started, I have 3 fantastic scientific co-founders, all of whom are academics through and through. So Jennifer Doudna and I were joined by James Berger and Martin Yinnick. James is now faculty at Johns Hopkins. Martin is a professor at the University of Zurich. All 3 of them have been in an advisory capacity at different times over the years. None of them wanted to jump in as full-time operators. So when we started the company, I was employee number 1 and the only employee for a period of time. So it was also a really rapid lesson in how important the team is and just how challenging it can be to find the right team at the very beginning.
You spoke about risk. What was your thought process about risk? Like, did you ever sit back and evaluate what is the kind of risk I'm taking and what are the consequences? I ask this because I think the biotech ecosystem, the startup space has evolved so much in the past 10, 20 years. It was different, I'm guessing, for you. I heard from Doug Crawford, finding space was an issue. It was so basic. What is that decision-making process for you like?
I think I was incredibly naive as to how challenging every element of this was going to be. And I actually see that as a benefit. I think if I appreciated how hard it was going to be, I may not have jumped in, to be totally honest. I think the enthusiasm combined with just the right amount of naivete gave me the chance to just dig in and quickly try to figure out how to solve one problem and how that could open the door to solving the next problem, so on and so forth. So no, I didn't have any sophisticated Excel spreadsheet where I was trying to rack and stack opportunities and risks. It was very much an enthusiastic jump in with both feet and figure out how to do this opportunity.
I think naivety helps. Naivety really helps. We try to warn people about how hard this can be. But moving along, I saw this tagline, persistence is a solution, in several of your presentations and your webpage. It looks like it's a theme for your therapeutics that you're developing and also your own journey. Such a simple but profound theme. We'll come back to this theme throughout our conversation. Maybe this is a nice thread to build the story of Caribou around. Let's get started about the technology itself. Give us a high-level description of the problems that you're solving at Caribou. Frame how complex the problems are, the existing solutions, and the kind of solutions being developed at Caribou.
I'll start by talking about technology, and then I'll bridge that into the kinds of product candidates that we're developing with that technology. At our heart, we're a CRISPR genome editing company. Obviously, given our roots, that makes a lot of sense. As we've been focused on the genome editing space for years, we've identified one of the key technology challenges as being specificity of genome editing. So what do I mean by that? These are biological proteins that we use to try to target the genome, and they inherently, at some frequency, target other sites of the genome. And we wanted to really push the technology to have far better specificity so that we could do a far better job of editing the one site that we intended to while avoiding off-target consequences elsewhere. This objective and many years of work here at the company ultimately led our team to invent a next-generation CRISPR technology that we call the chRDNA technology. So chRDNA is an acronym, CHRDNA, and it stands for a bit of a mouthful, CRISPR hybrid RNA DNA. So this describes the proprietary guides that we've invented here at the company. As I'm sure most listeners are aware, typically a CRISPR enzyme is targeted to the correct site by an all RNA molecule. What our team did instead was develop hybrid molecules that are part DNA, part RNA. And it turns out the inclusion of DNA dramatically improves the specificity of genome editing, hence the name CRISPR hybrid RNA DNA, or chRDNA for short. This technology, we think, gives us a real leg up in terms of making far more specific edits and ultimately being able to make multiple edits while maintaining genomic integrity. So cool technology, but what do we want to do with it? Today at Caribou, our focus is on cancer. And we are using the chRDNA technology to develop a wholly owned pipeline of off-the-shelf cell therapies, both for hematologic malignancies, liquid tumors, as well as solid tumors. And you're right, a key theme across our pipeline is persistence. So why do we focus so much on this? As we look at the cell therapy field, we're incredibly excited by the initial wave of work that's been done that has led to the approval of autologous CAR T therapies. So these are T-cell based therapies developed from a patient's own T cells that have been engineered in the laboratory to direct them to a particular protein on the surface of the cancer cell to kill the cancer. These products have saved lives. There's no question about it. It's incredibly amazing for the patients for whom they work, what these outcomes can be. I think we've also learned as a field that this is a really challenging approach to scale to the broader number of patients who could benefit from this. Because fundamentally, you have to manufacture a single batch for each and every patient. And we're seeing that's a major bottleneck today. So at Caribou, our focus has been on really advancing the field to the next level, which is going to so-called off-the-shelf or allogeneic cell therapies. There are a lot of compelling reasons why this will be better for a much broader patient population. However, there's an interesting technology problem that we feel we have to solve. That problem is the fact that off-the-shelf cells are foreign to the patient's immune system and therefore rapidly rejected. And it's this understanding that has led us to be so laser focused on using our genome editing to enhance persistence, with the goal then of being able to develop off-the-shelf cell therapies that have meaningful activity in the clinic.
That is fascinating. Thank you for that quick summary of the technology evolution. Let's go back to the beginning again. When you started the company, I'm sure you had so many problems that you can go after. What did you guys decide? Has it always been cancer or has it evolved? Talk us through the decision-making process.
Caribou today looks very different from Caribou on day zero. We've gone through a massive pivot as an organization. Winding the clock back to the beginning, you're right. There are just an incredible number of different opportunities where we believe CRISPR genome editing could be transformative. And we initially positioned Caribou as a pure platform technology company. We explicitly did not develop our own products, and we instead partnered with companies across a wide variety of market verticals in order to help them deploy genome editing to solve research problems or to develop new products. It gave us a chance to work with companies like DuPont Pioneer, focused on corn engineering, high throughput target identification, screening with a team at Novartis, and a number of other applications. This gave us access to capital that we were able to bring into the company, gave us access to technology, but I think more than anything, it gave us a front row seat to all of these companies who were already investing in using genome editing and the chance to ask them, what's the bottleneck? What are the things you want to do and why can't you do them? And that's actually how we learned that specificity was going to be so important to the field. And it was that feedback from so many of these conversations that led to the work that ultimately led our team to invent the chRDNA technology. And with that new technology platform in hand, we had the chance as a team to really hit the pause button and ask from a strategic perspective where we wanted to go with this exciting new CRISPR technology. And ultimately, we felt we had a real opportunity to do something for patients. And we pivoted the company. And today we're probably a far more traditional drug development organization. Our initial focus is oncology, as I've described. And we certainly see a lot of potential across other therapeutic areas in the future as well.
Speaking of pivot, how far along this journey did that happen since incorporation of the company?
We were a platform technology company for many years prior to pivoting into drug development. At that pivot point, we really had to take stock of what we were good at and what we were missing to then execute on our new objective of developing drugs. So at the time, we were really good at CRISPR genome editing, but didn't have a lot of people around the table who were really good at drug development. So we had to do quite a bit of team building to bring in the expertise to really translate the technology into therapeutic development.
We'll come back to these topics of this pivot and what investors said about those topics and how you build those teams. But I wanted to come back to your technology itself. You mentioned Chardonnay is far more specific genome editing than CRISPR-Cas9. Can you give us a scale of the improvement? Also, where you'd like this to be if you had your way?
That's a great question. And I'll say for those in the audience who really want to go down the rabbit hole on this, we actually had the opportunity to publish a manuscript with Martin Yannick and his lab, one of my co-founders, towards the end of last year, where in collaboration, we did a lot of biochemistry and his lab did some structural biology to really dig in on the specificity of the Chardonnay technology and understand the mechanism of enhanced specificity. We see that these Chardonnay guides reduce the rates of off-target editing typically by several orders of magnitude, to the point where we actually cannot detect off-target editing down to the limit of detection of the assays that we use.
That's a phenomenal improvement. And my background, I was working on RNAi delivery. Where does delivery stand today, and how do you see that?
Delivery is critical for so many of the applications of CRISPR technology in the therapeutic arena. The work that we do here at Caribou focuses today on cells that can be manipulated ex vivo or outside of the body. So for example, our product candidate that's in a phase 1 clinical trial today is manufactured based on healthy T cells from healthy donors. So we actually take those T cells into the laboratory and use electroporation to deliver the CRISPR enzyme and the Chardonnay guide to do the genome editing. There are obviously other therapeutic applications where you'd actually want to deliver the CRISPR reagents directly into the patient's body to specific cells or a specific organ. And that obviously requires a bespoke delivery technology. Much of what our field is doing today is borrowing from what's been done in RNAi. There's a lot of overlap in terms of the kinds of nanoparticles that have been developed historically and how they could be deployed for CRISPR genome editing today.
Wow, I didn't know that you were using electroporation. Like, I'm guessing it must be gentle enough that it's not affecting your clinical trials for that matter. Speaking of phase 1, Antler, can you talk about the disease area you're going after and that journey?
Our lead program is called CB10, and it's an off-the-shelf CAR T targeting a protein called CD19. And this is for the treatment of relapsed or refractory B-cell non-Hodgkin lymphoma. We are not the first to develop an off-the-shelf CAR T against this target. However, we are the first to make a very specific edit to the product candidate. We use our Chardonnay technology to get rid of PD-1. PD-1 is really a mechanism of brakes for the T cell. So by getting rid of PD-1, we take the brakes off the CAR T cells with the aim of keeping them in a high anti-tumor activity state for a longer period of time, really preventing their exhaustion through the PD-1 pathway. We saw in our preclinical studies in mice that the PD-1 knockout leads to a really significant increase in overall survival for mice treated with CB10 compared to mice treated with a nearly identical CAR T that still expresses PD-1. So in true Caribou fashion, we named our first phase 1 trial the Antler trial. And today we are evaluating CB10 in patients with multiple different aggressive subtypes of non-Hodgkin lymphoma.
And what other things that you have in your portfolio in the pipeline?
In addition to CB10, we have 2 other off-the-shelf CAR Ts that we're working on. Perhaps not so creatively, they are named CB11 and CB12. The first of those is for multiple myeloma, and the second of those is for AML. They are both in preclinical research stages right now. For the multiple myeloma program, we plan to submit an IND application to the FDA before the end of this year. And for the AML program, we plan to submit an IND application next year. In parallel, we're actually working on a second cell therapy platform. So everything I've talked about so far has centered on T cells and certain kinds of blood cancers. The other side of the organization focuses on natural killer cells, a different part of the immune repertoire, and their ability to potentially fight solid tumors. About 10% of cancer cases are liquid tumors. The other 90% are solid tumors, which we think represents just an incredibly compelling unmet need as we think about the potential power of cell therapies in that setting. Now, that said, there's been a lot of early work done by other groups over the past many years testing CAR Ts against a wide variety of solid tumors. And it's been pretty underwhelming. Many of them haven't worked well and many haven't worked at all. So our team really looked across the immune repertoire. We got really excited about natural killer cells instead because they inherently have anti-tumor activity. So we see that as a great starting point. And then we can introduce a number of different genome edits to really address many of the challenges inherent in solid tumor targeting. To actually do all of those genome edits, to get them in there, we start with induced pluripotent stem cells to manufacture these. So at the iPSC stage, we can introduce a multiplicity of genome edits, pick out a single cell clone, and then using proprietary protocols that we've developed here at the company, differentiate those edited iPSCs into natural killer cells with anti-tumor potential.
Fascinating. I'd like to go back to this concept of off-the-shelf therapies. Rachel, you made it sound so easy, but I can imagine how hard it is to build a universal solution like that. Can you please say more about this idea and what it means to a patient?
From the patient's perspective, and as I think about, for example, our ongoing ANTLER trial, for those patients who are on study, as far as they're concerned, their allogeneic CAR T therapy is ready the moment their physician decides they should be enrolled on the study and should receive a dose of it. So what we're able to do is really decouple the time and the effort of manufacturing from the timeline of the patient experience. Whereas today, when a patient goes to their physician and is receiving a prescription for one of the approved autologous or patient-specific therapies, it's their own blood that has to be taken, shipped off to a manufacturing facility. It takes, on average, something like 4 to 6 weeks for that turnaround time for them to ultimately get that therapy. Many of them are quite ill, so their disease continues to progress during that 4 to 6 weeks. They have to go on other bridging therapies to try to stabilize the disease during that time. It's very challenging. Whereas in the off-the-shelf setting, there is no wait time. It's literally in the freezer already. Now, that said, for the internal team, it's not like you snap your fingers and magically there's product in the freezer. And in fact, here at Caribou, we've invested heavily in building out our own internal process development team. So this means I have colleagues here at the company whose job it is to actually create the manufacturing process for how we manipulate the T cells. So they do everything from the earliest handoff from our colleagues in research to scaling it up, optimizing it. Ultimately, they tech transfer that process to a contract manufacturing organization who follows the process and makes the materials that we can then put in the freezer and utilize in our clinical trial.
That is so fascinating. This is almost like magic for people and a different way of thinking. Let's shift gears into the team. So you're a lonely team member when you first started, and I look at your webpage, you have a phenomenal team. How were you able to attract the best talent along this way? And what are the lessons learned from building your team? I read that your team retention is higher than other biotech companies in the Bay Area. I'm guessing you're doing something right there. Talk about the team.
I'd say one of my early learnings building Caribou was just how important the team is. Perhaps coming into all of this, I assumed the most important ingredient was the technology, but I now feel that was wrong. You can have the coolest technology on the planet, but if you don't have the right people sitting around the table, it really doesn't matter. Especially in the early days, it's really hard to build a team, especially when you don't have enough money in the bank account to pay attractive salaries. So it certainly helps at that point to have a great technology in order to entice people to get excited about the opportunity and to jump in. I have to give a lot of credit to my founders and Jennifer Doudna in particular, who helped open a lot of doors early on to meet people in the network who were willing to do anything from sit down with me over a cup of coffee for an hour to give advice through to ultimately joining the team as a full-time employee and helping to really guide the future of the company. I think the first 10 or so hires are the most terrifying hires to make because each person has such a profound influence on the company, both in terms of the direction of the company and what you do on a daily basis, but also the culture of the organization. And I will readily admit that coming into this as well, I thought culture was one of those buzzwords that you read in business school textbooks, but didn't really matter. I could not have been more wrong on that front. And I'm really grateful to one of my early colleagues who came in with a very clear mission and vision personally on how to build and shape the culture at Caribou. And I think the organization benefited tremendously from someone early on pounding the table on that front. And it continues to be an area of focus for us today.
Speaking of culture and the mission and vision, when did you actually realize that this is really important and you started taking it seriously? And from that point on, how did that evolve? What was your perception about this is how my culture should be? And who did you learn from other than this colleague of yours? Because a lot of startups, like you said, we don't focus too much on the culture and the company building process because we love our technology scientists. We do that. And I think we are trying to change that. Lessons learned will be very helpful.
I gleaned an appreciation for the importance of culture fairly quickly, thanks to this early colleague. And it meant we spent a lot of time talking about it, but we didn't actually write it down for many years. I think that's probably fairly normal. There are a lot of early stage companies who benefit from the fact that communication's actually really easy when you're a small team. You can put everybody in one room, you all sit there and have lunch together, you have a single conversation and everyone's on the same page. Very quickly, as the company grows, that is no longer possible. So finding ways to codify, to clarify culture, mission, vision, values becomes incredibly important so that everyone has the same source point for what is true north, what are we trying to do. I think one of the hard things is it continues to evolve. As the company continues to grow, practices for communication change and change again and change again. It's one of the things that continues to keep me up at night. We're at a point where we're about 120 people or so. So some of the communication mechanisms we used 2 years ago or even a year ago really just don't work as well today as they did then. And it's really important to continue to figure out how to evolve these strategies so that we all remain on the same page and have access to the information we need to effectively do our jobs.
Can you please say more about how you arrived at the mission and values of Caribou and how it has evolved over time?
Several years ago, we ran our first, I'll say formal process, but maybe I'll put air quotes around the word formal, to articulate our corporate mission. As time goes by and what the company focuses on evolved, we realized, again, the mission no longer really aligned with actually what we're trying to do as a company today. So again, last year, we had the opportunity to really reframe the mission and the values of the organization. I ran a process where I invited people from across the company to join me. So it was close to 10 people representing many of the different groups at many different levels of the organization. We took a lot of feedback and ideas from our colleagues across the company and ultimately landed on a single mission statement that we now put everywhere. It's on our corporate deck. You can find it in a lot of materials. In fact, most of us walk around with little laptop cases that have the mission and the values printed right on it. I think that's something that makes a ton of sense for an organization of our size, where, again, we can't all just sit around one table at lunch and have one conversation and be on the same page. It's a great tool for making sure we can all point back to why we're here, why we put in the incredible hard work that we do, and how we come together as a team to successfully get the job done.
Speaking of having different conversations and ideas, like you have a phenomenal team and they all must be having diversity of opinions. How do you process them? And how do you decide whether something is good or bad? And how much does your gut play a role in this?
I feel incredibly lucky that I get to work with so many people who have incredibly deep expertise in a wide variety of areas. And I think we're successful as an organization because we're able to get people in the room who have not only a diversity of backgrounds, but a diversity of educational expertise, work experience, and subject matter expertise. I think it's incumbent on us as an organization to figure out how to integrate all of those different viewpoints and ideas, but still at the end of the day, make decisions in a timely fashion. A former colleague of mine used to warn against what she called analysis paralysis, which fundamentally boils down to making no decision is worse than making a mediocre decision. We're in an industry where technology moves quickly. Patients need solutions yesterday. So not making a decision is probably the worst outcome for the company.
Do you ever second judge your decisions once you make a decision? And what is that process looking like? Who do you talk to?
Nothing is ever a straight line. There are plenty of decisions that we have made that we go down a path and ultimately realize, okay, this probably wasn't the right path. What do we know now that we didn't know then? How will that help us make decisions like this again in the future? And then how do we get ourselves out of this and into the next solution? There has to be an iterative process for so many things that companies do, but again, not to the point of paralyzing the organization. It has to be a balancing act of, we all sat in the room, we all worked together, we came to a conclusion. We said, go to X, Y, and Z. We're going to go do X, Y, and Z. But if new facts come to light or we're not as successful as we thought we were going to be, there has to be an opportunity to revisit the topic and question whether we keep going in spite of the difficulties we're facing, or do we need to hit pause and go back a different direction? I think everyone's gut has to be part of that. All the people around the room who are part of the team working on it, but there has to be some structure to it as well. I think we as a company are continuing to build some of that infrastructure for codifying how decisions are made and then how we all get aligned and take next steps.
You need to have a phenomenal team with phenomenal guts to get to some good decisions. Speaking of that, is there a process that you have while you're picking somebody to join your team that worked for you? And if there is something that didn't work consistently, if there is any lesson learned, that will be helpful.
Hiring is so hard. Even today, I think it's one of the hardest things our company has to do. And personally, when I'm involved with helping to hire someone, I think it's one of the hardest things I have to do. And I think that's because you typically only have a fairly short window of time to interact with someone who you've probably never met before. You're trying to suss out what skills they do or do not have and whether they're going to fit in with and enhance the team or disrupt the dynamics of the team. I'll say for me, one of the most impactful elements of the recruiting process is actually the reference checking component. I personally do the reference checks for most people whom I'm hiring. I know often when people hire a recruiter, they just ask the recruiter to do all the reference checks for them and they read a report and figure out what to do with it. I fundamentally believe that is a unique window into this person. It is a chance to really understand from people who worked with them or for whom they worked for many years, how they show up, how they function, what their skills are, what they're still learning how to do, and how all of that overlaps with what our team and our organization needs at this point in time. It's a tough process, but one that's obviously mission critical to the future of the company.
What is one question you ask during those reference checks that is consistent and that helps you decide if the other person is actually giving you a good answer, for lack of a better word, the right answer?
I love asking the question, would you hire this person again? And every once in a while, you get the answer of, I did. I hired them twice at 2 different companies, and here's why. And that's the best answer to that question. Many others would tell you, yes, and here's the kind of role I would hire them for and why they're not at my current organization because we didn't need that role. And then others try to dance around the question, and that's pretty telling.
That's a nice segue to talk about your leadership. How did that evolve? Because you didn't come with an experience as a leader from grad school. How did that evolve? And what are the lessons learned in that process?
Tremendous amount of learning on the job. I have tried to learn from my colleagues and my peers, but I've also been quite proactive about joining organizations or taking advantage of opportunities where I think I can learn. That's a mixed bag. It's everything from working with an executive coach, which I have done for years, to participating in an organization that is specifically for the professional development of CEOs. There's a small group that I'm part of. It's about maybe a dozen of us who get together half a day a month and share some of the strategic challenges we're facing and learn from each other ideas for how to solve those challenges. That's been incredibly impactful, through to actually joining the boards of other companies where hopefully I have something to contribute and bring to that company. And I also know that I learn a lot from seeing how another company, another leadership team, and another board functions and tackles many of the same challenges that we face as well.
Before I move on, I'll be amiss if I don't ask about Jennifer Doudna and the role she played in the company building process. Do you feel more stressed after she won the Nobel Prize that you got to make this work?
I'll say the timing of her Nobel Prize was spectacular, selfishly for Caribou. It was shortly before we kicked off our Series C financing. So it really doesn't hurt to go knock on investors' doors and get to say, my co-founder, Nobel Prize winner, Jennifer Doudna. But winding the clock back all the way to the beginning, Caribou is actually the first company that she helped to co-found. I think she and I went through a lot of learning together. She was incredibly enthusiastic. I think it's fair to say remains incredibly enthusiastic about the potential of the organization. And I think it's fair to say she's very excited about our recent clinical data that we shared just recently. Even at the time, in the infancy of CRISPR world, she was already a very well-known scientist in her field and had a Rolodex in Silicon Valley that she opened up to me. She was able to get me into the room for a lot of initial conversations that were critical to hiring people who ended up being on our team for years, finding potential partners, and ultimately raising our first round of capital as well.
That is a blessing to have somebody like that as a co-founder. I'm jealous of you. I'm glad you had one. Speaking of money and fundraising, maybe you can talk about your fundraising evolution from your seed to Series C and then going to IPO. You talked about you were naive when you started the company. Did you have any ideas about this fundraising, like what it was going to be and how it evolved? Let's split this into 3 stages, from your idea to your seed stage, and then the venture capital rounds, and then going IPO.
The very first money that we brought into the company was a combination of friends and family who were willing to write some checks to help us open the doors, and 2 SBIR grants. So I wrote and got funded 1 SBIR from the NIH and 1 from the NSF. So that was just enough to get started. And then we really in earnest began conversations with venture capitalists. So thanks to Jennifer and a few others in the network, I was able to take first meetings with a number of the VC firms you've heard of. This was so early in the world of CRISPR that no one really appreciated yet how much all of this was going to explode. And most of the conversations ended with the investors not believing that there was a broad opportunity and saying, when you figure out the one thing this technology can do, give us a call, we'll have a conversation. So I'll admit I didn't think fundraising would be easy, but I also didn't think everyone was going to say no either. That bruises the ego pretty quickly. But at the same time, we were actually getting cold calls from DuPont Pioneer, one of the largest agriculture companies on the planet, and from Novartis, one of the largest pharma companies on the planet. And it's because their technical teams had read the paper out of Jennifer's lab. They saw the writing on the wall and saw incredible potential and promise for the future of this technology. We ended up going a slightly different path in terms of our Series A, and that first round was actually led by DuPont Pioneer rather than by a more traditional institutional investor. Novartis participated as well, as did other institutional investors. It gave us a path to capital raising that wasn't even on my radar when we first got started, and ended up being an incredibly beneficial start for the company. I'll admit, I think we fell victim to a set of challenges that probably a lot of founding teams do, which is agonizing over the valuation of the raise, how much money you raise, how much dilution you face. At the end of the day, it's all a little bit silly, because if you own a big piece of something that's not worth very much and not going anywhere, that's not very valuable. So I'd say one of the lessons I learned early on was raise more money than you think you need. You're going to need it. Don't sweat the details in terms of dilution and valuation, because at the end of the day, if you're really successful, which requires sufficient capitalization, everyone's going to win. I think that's a lesson that I've certainly taken forward. So as I look at each of the raises that we did as a private company, did a Series A and then a Series B, then a Series C, and then last year took the company public, I think we've integrated some of that learning of really raise as much as you can when you can, because you never know what's coming next. So thanks to the combination of our Series C, a partnership deal that we did with AbbVie, and our IPO last year we raised a total of $495 million. And given where the capital markets are today, I'm pleased we did that.
Congratulations on that. Give us a sense of the scale of funds raised, including IPO. I asked that question because I think early stage scientists like grad students underestimate how much money is actually needed to get where you are to the phase 1 clinical trials. Last year alone, you said $495 million. Talk about that subject. One thing you mentioned about dilutions and the valuations that early stage founders worry about. I would like to hear a little more on that subject.
So our seed capital was fairly modest. We initially raised maybe a few $100,000. I think we eventually got to about $1 million or so prior to our Series A. All things considered, a pretty small amount of money that facilitated only a pretty small team initially. Our Series A ended up being about $11 million. Our Series B was about $30 million. Our Series C was north of $100 million, and our IPO was $350 million. Now that we're a publicly traded company, one of our obligations is to share our financial information every quarter. So if people are bored, they can always dig into those filings. As you look across companies, you can really quickly gain an appreciation for just how expensive drug development is, especially once you're in the clinic. And it really requires not only tremendous expertise on behalf of the team, but also access to significant resources to be successful.
Talk us through the decision-making process of like you had a chance that you could go and raise more venture capital money as successful as you were until that point. And why did you go the IPO route?
I think probably every company comes to this decision for a few different reasons. For us, it was really about thinking about all the additional capital we were going to need to raise in the future and figuring out how much more we might be able to raise as a private company and at what valuation, and how much more we might be able to raise as a public company instead and at what valuation. We felt we'd really hit that inflection point where we had somewhat maximized our ability to raise capital as a private company, and that our next best option for the total amount of money we needed to raise, not only then, but going forward, would be as a publicly traded company. And certainly the largest round that we've raised to date by far was our initial public offering.
I asked that question because my assumption is that as a private company, you can take more risks, but as a CEO of a publicly traded company, you have to be more circumspect. What would you do differently if you had your way?
I would say I described our first 9 years as being blissfully private. You're absolutely right. As a private company, you obviously have shareholders who care deeply about the performance of the company. However, it tends to be a fairly small group of highly sophisticated shareholders who you've probably allowed to invest in the company because of their expertise and maybe even their ability to help build and grow the company. And it's a lot easier to have a conversation with them of, hey, we thought we were going to do A. Turns out A wasn't a great idea. We think B is a better idea, but it means we're not going to have clinical data until 2 years later than we thought. Now, they might not love that, but often there's a lot of support for continuing to evolve the strategy. As a public company, you're doing all of this in the spotlight. You're making promises to Wall Street about what you're going to do and when you're going to do it, and you get punished if you don't. So I think for any company that makes that transition into the public markets, one of the things you have to get really good at quickly is prediction. You have to accurately predict what you're going to accomplish and when, and stick to it. I think that's a new skill for a lot of companies.
That's a tough one too. Speaking of investors, maybe that's a good segue to talk about your board members. How did the board evolve and the role they played in helping you guide, and how did you pick those investors? I'm guessing you had to make choices of who to take money from.
We actually built a board of directors before we ever even raised the Series A. And I'm so glad that we did. It was in part because this is my first go round, and I felt I would benefit and the company would benefit from having more experienced leaders in the boardroom with a fiduciary responsibility to the company who could help shape even the first round of capital that we raised. It was a small board. There were 2 other people initially who we recruited. each of whom had distinct areas of expertise in industry who really helped us build and grow the company. Now, over time, the board evolved. And of course, we started to have some of the investors who participated in different rounds participating in the board of directors as well. One of my own personal philosophies all along has been that life is short. You should really do everything you can to work with people who you respect, and in the context of the company, who can really help the future of the organization. And that absolutely helped shape some of the decisions we made about who invested in the company, because we knew that that would also reflect on who joined the board of directors. We had a somewhat unusual situation last year. As the company was getting ready to go public, we learned that all of the investors who were then serving on our board of directors work for funds that don't let them serve on publicly traded boards. So they all had to resign either ahead of or at the time of the IPO. So it ended up being a really remarkable opportunity to almost rebuild the board from scratch. There are two of us today who were on the board prior to all of this, who are still on the board now. And it gave the team and me the opportunity to really envision an empty boardroom and think about who are the kinds of people we want filling these seats, what areas of expertise, what background, what roles they perform either today or prior to their retirement. So it's been a process. We started bringing on new people in April of last year, and our most recent new board member just joined us last month.
That is a fascinating story. Brand new board of directors and a new vision, different ways they can help you go there. Speaking of boards, I know that you're serving on other startups on the boards as well. What are the lessons you learned from your own startup journey? And what are you learning by sitting on other boards?
It is a wonderful learning opportunity for me. So today I sit on the boards of two other companies. One is a very early stage, still stealth organization. I'd like to think that some of my earliest experiences here at Caribou are beneficial to that company. And likewise, I learn a lot simply by seeing how they think about building and growing their nascent team and the tools that they use to communicate with each other and build and grow their company. On the other end of the spectrum, I also sit on the board of Seer, which is a publicly traded company in the proteomics space. I learn a tremendous amount by seeing how that leadership team navigates many of the same challenges that my team faces. Again, how that board navigates many of the challenges that my board faces. And I also get to learn fundamentally new things from that experience. For example, they're in the tools space. So they actually sell products to customers today. Here at Caribou, our lead program is in phase 1. We're not selling anything to anyone right now. So it's been eye-opening for me to learn how another company that has so much science and technology at its core is facing the challenge and the opportunity of actually commercializing its initial products.
Have you seen mistakes that early stage founders do in terms of composing your board of directors? I ask this because most scientists don't ever think of those and the consequences it might have as the company grows.
I think often early stage founders ignore building a board until they've raised money from investors and are forced to build the board. And at that point, the board ends up being pretty much all VCs with maybe the CEO, maybe one other person. So you end up with a bit of an imbalance where the VCs control the vast majority of the seats in the boardroom, and the founding team has maybe one seat, and maybe there aren't even any independent directors who are not affiliated with funds. I think that means you're missing important viewpoints in the room. It's not that any one group should own 100% of it. I think there's value in distinct viewpoints and areas of expertise. So I'm really thrilled that we actually took the trajectory we did, where we started with a board that was only independent directors initially, plus me, and then we added investors later, and always throughout had a pretty meaningful balance between investors versus non-investors on the board of directors.
I love this idea of building independent boards even before you raise money, which I've studied a lot about and I'm fascinated by. I think independent board members absolutely provide valuable points of view that may be different from what an investor might bring to the table, and also help you learn and grow tremendously, especially if you are not too experienced. Did the investors that came afterwards have an issue with having a board even before the investors came on? How was that experience like or conversations like?
I wouldn't say any of them at least shared with me that they had an issue with it. And in fact, I'd like to think that for some of them, it was one of the strengths of the company at the time. I recognized the fact that I was 26 when we founded the company and had no experience as a CEO was a risk, as some of these investors were looking at the company. And I'd like to think that as they saw experienced professionals on the board, that was a real vote of confidence that those people were voting with their feet to spend time with the company, but also able to help the organization.
I'll go back to talking about partnerships and companies. You work with DuPont, Novartis, and AbbVie now, where initially probably they were helping you in terms of by offering money, giving projects, and then now I think AbbVie, you're helping a big company, if I'm right. How has working with big companies been? Any lessons learned there?
It's a great question. And I'd say probably true for so many biotech companies. Partnership with larger organizations is a critical element of building and growing the company. It's one of the many ways that we can raise capital or gain access to expertise or technologies that we otherwise don't have. Interesting pros and interesting cons as well. As I think back to the very beginning of Caribou, as I mentioned, DuPont led our Series A, and it was a fairly modest investment, all things considered. And yet they had a governance, a decision-making process internally where any deal, regardless of its size, had to go all the way up to the CEO of DuPont, who had to personally approve it. What was otherwise a fairly simple decision took a really, really long time. In Caribou years, at least, it took a very long time and actually made some of the early days of company building challenging because our plans for when we wanted to hire people were far more aggressive than DuPont's timelines for actually greenlighting the deal. One of the pros, though, of having a company and not a traditional investor lead the raise is that companies typically, for various accounting reasons, don't want to own more than 19.9% of small companies. Because if they own 20% or more, the accounting gets fairly complicated in ways that is not beneficial to the big company. So unlike a traditional VC who wants to own as much of the company for as little as possible, the corporate venture capital group thinks about valuation differently. In fact, often their primary objective is strategic, meaning they're looking for companies that have technologies or area of expertise that they want to have access to. And their secondary objective is a financial return on investment. So that can often be a lot more founder friendly in the early days.
You were this young entrepreneur. How was that table looking like at two ends of the conversation? One is this big corporation. Who was on your support system? What kind of support did you get and guidance did you get? I'm sure you're lawyered up. Talk us about that conversation and decision-making process.
Almost always a pretty significant imbalance in terms of the sheer number of people on any call or meeting, that's for sure. But no, we quickly had to find the right areas of expertise on our side. So that was people inside the growing company who had the right scientific and technological expertise. That was finding initially attorneys outside the company who could help support the negotiation and a number of the other resources. Simple things like you need a bank account for the company. A lot of the other infrastructure that certainly as a Ph.D. student, I hadn't learned how to incorporate a company or how to open a bank account for a company or things like that. And that's actually something where the QB3 Institute played a really important role in the founding of Caribou. We were one of the first companies to go through what they now call the Startup in a Box program, where we got plugged into introductions and infrastructure to open doors to many of these different kinds of service providers.
Doug Crawford did a great job with the Startup in a Box program, and he's the one who actually introduced me to you. Thanks to him, and thanks for taking this invite to be a part of this. Startups are risky. That's an understatement. Did you ever worry about failure? How did you deal with that thought that you might fail? Can you speak about the coping mechanisms and the support systems you created for yourself when in self-doubt? Who were those believers to help you be propped up again and just keep going at it?
Of course. I think you probably have to be crazy to never face situations where you feel like you're on the precipice of failure. It's a really interesting tension between being persistent, to your point about that theme earlier, and just continuing to push forward, but not to the point of crazy. At some point, you have to listen to the external feedback and really figure out you're headed in the wrong direction. Is there a right direction? And if so, how do you pivot to get there? For me personally, it was everything from other leaders within the company who we could sit down and hold hands and figure out, was this just a tough situation and we hadn't found the right investors yet? Or were we really fundamentally headed in the wrong direction and needed to do something dramatically different for the future of the company? Through to friends here in the Bay Area who are entrepreneurs themselves and have been through the ups and the downs and the ups and the downs to help normalize that that's just part of the journey. I think that makes the United States, but the Bay Area specifically, really special on this front. In the early days of Caribou, I also spent a decent amount of time interacting with some entrepreneurs from the United Kingdom in the synthetic biology community. The culture is just completely different there. At least as they shared with me at the time, there were a lot of people who were nervous to be entrepreneurs because what would happen if you failed? Where would you possibly go next? Whereas here in the Bay Area, failure is almost a badge of honor. Now, you better have learned something, but it leads to new open doors and new opportunities.
Yeah. Bay Area is different. It's magic in the startup world. I'd like to move from teams and your company to you as an individual. I read in one of your articles as I'm very comfortable with being underestimated. You can knock it out of the park while everyone else is looking the other way. That's a quote from the book Girls Who Run This World by Diana Kapp. You've absolutely been hitting it out of the park. As a female founder, were there challenges that were unique to you from a gender perspective? And how did you overcome those issues and speak of this support system from a point of view of like if any other female entrepreneur is going through this journey, like what is it that they can do to help themselves?
There are so many times over the past 10 plus years where I've been in a meeting outside of Caribou and I've been the only woman in the room. I think that there are actually fairly few times where I feel I've been mistreated because of my gender or mistreated because I was the only woman in the room. There, of course, have been a few, but not a lot. I actually think more often I've been overlooked or underestimated because of my age rather than my gender. And that's where that quote came from. I fundamentally realized being overlooked could actually be a real silver lining. It meant that people weren't staring at us constantly and we could quietly put our heads down, do amazing work, and come out the other end having earned the credibility by doing what we said we were going to do. Fundamentally, our interactions with investors are incredibly relationship-based. And by and large, they start with us telling them what we're going to do. And we disappear for a while. We do it. We come back, show them we did it, and tell them what's going to come next. Because I don't have 3 other companies that I've run before that I can point to say that's my track record. I personally, and we as a company, have to develop that track record here. Now, inside the company, Caribou looks really different from most other biotech companies. Across the organization, we're at gender parity. Our board of directors has more women than men. I don't know the stats, but I'd hazard a guess we're one of the very few publicly traded companies for which that's the case. Our executive leadership team is half men, half women. I think our gender diversity has actually been an incredible advantage for us in terms of recruiting. We have heard from job applicants that when they interview with us, they see people who look like them. And when they interview with other companies, that's not been the case. So I think we've been able to tap into a talent pool that has been overlooked by other organizations. Our field is evolving. There is work yet to be done. Still at the C-suite level and the board level in biotech, there's a pretty significant imbalance. It's better than some other industries, but it's not where it should be. I think there's a lot of opportunity for many of us to continue to be in a situation to lead by example and demonstrate that we belong and we have a role in a space in this industry.
Congratulations on building such a diverse team. Like I looked at some of the pictures, I was like, this must be intentional. Moving along, you must be extremely busy running a public traded company. How do you balance company life and your other industry commitments? How do you stay healthy physically and mentally?
I'd say years ago, I realized I actually had to be a lot more proactive about trying to achieve some sort of balance between life and work. to the point where I actually calendar my runs. So I love running. I'm very slowly working on running a marathon in each of the 50 states. And I finally realized that if I don't actively prioritize something, including putting it on the calendar, it doesn't happen. So I've realized that I'm a far better person. I'm a far better leader when I'm healthy. And that includes eating real food, sleeping sometimes at night, and exercising. I've done, I'll say, better now than I used to at actively prioritizing those things, because I know that I can't be who Caribou needs me to be if I'm not taking care of myself.
Prioritizing is really helpful. Easy to get busy with work. My podcast is around lab to startup. We're trying to encourage more of grad students and postdocs to take their research, start companies. I'm trying to normalize company formation. I think the way academia normalized becoming a faculty, I think there should be an effort entrepreneurship. Any advice for those sitting on the fences? I get to hear this all the time, that they're scared. Any advice will be helpful.
I'd say there's so many tools to learn. I was lucky that when I was a PhD student at Cal, I had the opportunity to enroll in quite a lot of business school classes while I was there. I ended up with a certificate in management of technology. Certificate is neither here nor there, but it was really about the opportunity to learn the vocabulary. The phrases you use when you're negotiating a Series A sound nothing like the phrases you use in a lab meeting when you're presenting your thesis research to your colleagues. So it was a great opportunity for me to do some of that learning ahead of time and then have that vocabulary ready to then walk into those rooms and learn so much of it on the job. Don't get me wrong, but I at least had the vocabulary to get started. So I would encourage people, whether it's formal classes or pick up a book, watch some videos on YouTube. There's so many ways to learn about organizational behavior, company building, accounting, and finance. You should absolutely take advantage of any of those opportunities. I think another key learning for me, and something I feel I learned from Jennifer Doudna very early on, is the importance of storytelling. At the end of the day, company leaders have to be good storytellers. And one of the critical elements to being a good storyteller is knowing your audience. You might be talking about the same underlying technology 3 times in one day, but one time you're talking to your internal research colleagues, one time you're talking to an investor who is an MD by training, and one time you're talking to an investor who's an economist by training. You're going to talk about the same topic in 3 really different ways with those 3 different groups. And I think learning how to get comfortable having those conversations, telling those stories to different audiences is incredibly important at the beginning of an entrepreneurial journey.
Thank you for that great advice, Rachel. Before I end up with my closing questions, CRISPR, I think it's still in the early stages. Where do you see this 5 to 10 years from now? And where would you want to see it to be in terms of solving the problems? I also want to tag along on the question about, are there any complementary technologies that need to evolve to prop this up? I think there's a great need. It's a powerful technology, but it's not there yet.
I'll start with where is Caribou today? And then I'll share where I hope the whole field is 5, 10, 20 years from now. So today, Caribou is at the point where we shared our very initial clinical data from our first clinical program. So a really exciting inflection point for us. We shared outcomes for the first 6 patients that we dosed with CB10. And quite remarkably, at only dose level 1, which is a very low dose for this kind of cell therapy, we saw a 100% complete response rate, meaning each of those 6 patients responded to CB10 and in fact had no detectable cancer as their best response to CB10. The very first patient to receive CB10 is now a year out, more than a year out from receiving his single dose, and he's still in complete response.
Wow, congratulations on that. Big deal.
Thank you. It is a really big deal, and I think it helps the team and it helps me find a number of really great reasons to get up in the morning and do the hard work that we do, because we know that the blood, sweat, and tears of many years have actually helped these people and their families and show such tremendous promise for what can be done in the future. So what does that future look like? Today, by and large, medicines available to people fall in 2 categories: small molecules or the category of antibodies or other proteins. So I believe in the future, we will have 3 categories, those 2 plus what I'll call genetic medicines. And that would include genome editing, cell therapies, RNAi, other ways where you're directly manipulating the genetics to treat disease. Obviously, there's a lot of work to do to get from where we are today, which is a lot of preclinical research and a handful of clinical trials, to a robust number, hopefully, of approved products for these patients in the future. But I think it's also really clear that human therapeutics is not the only field where CRISPR genome editing has incredibly broad potential. I talked earlier about some of the collaborative work we did before with DuPont Pioneer. They are obviously very interested in corn and other plants and solving challenges genetically using genome editing. Caribou actually has a number of licensees today who are using CRISPR technology in a wide variety of applications, whether it's trees, pigs, cattle, corn, or otherwise. And I see a lot of potential on that front. It's a really exciting moment as we think about the next 5, 10, 20 years and what's possible. To the last piece of your question, I think one of the critical elements to unlock a broader set of opportunities within human therapeutics is a topic you brought up earlier, and that's delivery. There are many genetic diseases where you would want to be able to edit the genome of a very particular organ and probably no other organs. And we're just lacking in the robust delivery tools to get to most organs today. Today, there's some pretty exciting early work happening in the liver and in the eye, but there's a tremendous amount that needs to be done. And I'm hoping maybe some of the listeners are investing in this and inventing technologies in this space to help unlock even more of these opportunities in the future.
Congratulations again, all the success you had in painting that picture of the future, and hope there'll be more entrepreneurs like yourself helping build this ecosystem. Are there any topics that I haven't touched upon about your own journey with Caribou or the company that people should know about?
I think you've hit a lot of the highlights. I'll just end by harping on one of my favorite topics, which is team. At the end of the day, it's really all about the team and the amazing people who enable what is possible.
Thank you for that. And now to my closing questions. First one, are there one or two lessons that you learned from your own mistakes that others could avoid?
I'll highlight one that I shared earlier, which is really about fundraising strategies. And I would encourage anyone to raise more money when they have the opportunity to, even more than you think you need. Nothing is ever straightforward. This is science. It's messy biology. It's very hard to predict. And I think under-resourcing a company only limits its opportunities. That's been a key learning for me. If I could go back again, I'd like to think that I would have raised larger amounts of capital earlier on.
That's a great point. I still underestimate how much money I will need to raise, but I think we need to hit on this point again and again. Thank you for that. And my second question is, are there one or two people or more that have played a significant role in your startup journey that you'd like to acknowledge?
Oh, absolutely. I'll start, of course, with my co-founders, Jennifer Doudna, Martin Yannick, and James Berger. Without them, the company wouldn't exist. And I'm incredibly grateful to them for giving me the opportunity to be the founding CEO. To this day, I'm still not entirely sure why they were comfortable with a 26-year-old who had no experience being the founding CEO. But not only did they give me that chance, they continue to give me their support. And I'm incredibly grateful to them for that. And then since then, obviously, we've had so many fantastic people on the team. It takes a village, especially at an inflection point that we've had recently of sharing our initial very promising clinical data. It's been a wonderful chance to think back on literally every person who has contributed to that and all the help we're going to need going forward to continue driving this program and other programs forward.
Rachel, thank you. This has been a terrific conversation. Thank you for sharing your journey and the story, and I'm hoping this will inspire a lot of other entrepreneurs. Thank you for being on the show.
Thank you. So appreciate it.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.